Now let's get started. The first thing you want to do when you come to the lab, you need to make sure that the equipment that you'll be using to manipulate cells with is sterile. So let's see how we sterilize different things. First of all, when you enter the lab, please check the settings of the equipment that you'll be using. Check, for example, the incubator settings before you place your cells in them. Most incubators are set at 37 degrees Celsius, so make sure that you're using the appropriate incubator. Also, these incubators, as I have mentioned earlier, have water tray in them, water container that keeps the environment humid. It's 100% humid environment. Also, if you are using bicarbonate media, make sure you're using incubator that has carbon dioxide function in it. Generally, different cells have different requirements. Generally, people use 5 to 10% carbon dioxide for different types of cells. So make sure you are using the correct incubator. As I mentioned earlier, bicarbonate maintains the pH of the media. Some cells can be grown in media that is not dependent upon carbon dioxide. So they'll be placed in a different incubator from the one people use to put cells in, which requires carbon dioxide for maintaining the pH. So once you have checked the settings of your equipment, you will need to sterilize different equipment that you will be using. There are different strategies to sterilize the equipment. Let's talk about them one by one. UV, ultraviolet light. Ultraviolet light is basically electromagnetic radiation, and if it is of sufficient strength, it can cross-link different molecules. For example, it can cross-link two thymines in the DNA. So if DNA is damaged, the microorganisms will not be able to propagate. So this is one way you can kill different organisms in the area that you need to work in. For example, UV light is present in the laminar flow hood. Before you start working in the laminar flow hood, you need to turn on the UV light. You can also use UV light to sterilize other pieces of equipment. Ethanol is a very useful agent to sterilize samples. 70% ethanol is the most effective concentration. It basically can enter cells easily, and it can also precipitate different proteins and kill the microorganisms that you want to get rid of. Baking. Baking basically kills microorganisms by heat. So different labs would use different temperatures to bake their material. For example, glass is generally around 300 degrees Celsius. Autoclaving, basically certain reagents or certain material that you cannot bake. For example, you cannot bake plastic because it will melt. You cannot bake media that you have prepared because if you do that, it can denature the different components in the media. So for such pieces of equipment or such reagents that you want to sterilize, you can autoclave them. Autoclave basically kills germs by two different mechanisms, heat and pressure. So autoclaving is also a very good strategy to kill different organisms. Flaming is also very important. So when you're working in a laminar flow hood, when you open your sample tubes which have reagents in them, and you want to make sure that nothing which was present around the lid enters the sample, you can flame it. When you flame an object, it also destroys the microorganisms. It is not because of higher temperature, because the exposure to flame is very, very brief. The killing is basically a result of oxidation. So basically, when you burn something, burn a microorganism, it will, of course, become non-functional. Filtering. A sensitive media which cannot be autoclaved, we can sterilize our media using different types of filters. We will talk about the filters in the next slide. Filters are like a membrane. They have little holes in them. So when you pass a material through a filter, filters have different sizes of holes. Generally, these are small enough to restrict the movement of bacteria or other pathogens through those pores. So when you filter something, the particulate material will stuck on the filter, whereas the liquid, the solution, and the solvents material present in the solution, they will easily pass through those pores. Generally, we use pores for the pore size we use for sterilization is 0.2 micron. There's another type of filter which has a 0.45 micron pore in it. 0.2 micron filter is used to remove bacteria from tissue culture media. Smaller particles, for example, mycoplasma and viruses, they can actually pass through 0.2 micron filter. 0.45 is primarily used to remove particulate substances from solutions. So if you make a solution and you see there's particulate material in the solution and you want to get rid of it, so you can use 0.45 filtration system, because of course it is relatively cheaper than using the 0.2 micron. Use an appropriate filter size and type. Of course, there's one of them is, for example, cellulose polycarbonate. So this is another way you can sterilize things. Now let's see what is the preferred material of sterilizing different material. As I mentioned earlier, media should be filtered to prevent deactivation of important molecules present in the media, because other alternative, of course, would be to either expose it to the UV light or extreme temperature. You don't want to do that. So media should generally be filtered. Tissue culture hood should be cleaned with 70% ethanol. And again, after you have cleaned first of all, you turn on the laminar flow hood or tissue culture hood. Once the air jet starts running, you clean it, wipe the whole thing with 70% ethanol, and then you leave the ultraviolet light, which is also part of the laminar flow hood or tissue culture hood. You leave it on for about 15 to 20 minutes, and once you have done that, you can turn off the ultraviolet light, and of course you can use the regular light and you can work in the laminar flow hood. Plastic, as I've mentioned earlier, can be autoclaved, UV treated, or plastic can also be sterilized using by spraying 70% ethanol. Glass, of course, can be autoclaved, UV treated, sprayed with ethanol, or it can also be baked, because you don't have to worry about glass melting when you bake it. Metal can be autoclaved, UV treated, sprayed with ethanol, or metal can also be baked. In order to quickly sterilize things, we can use the flame inside the laminar flow hood. There is a gas connection to which there is a burner attached. So whenever you open a tube, a sample tube which contains media or whatever reagent, you want to keep it sterile. When you open it, you sterilize it, and after you have removed the materials you want to move from the tube, or you have added some material to the tube, you again flame it, both the mouth of the tube or the container and the lid, and you close it. So this is basically how you sterilize different pieces of equipment or reagents. So once you have sterilized your equipment, your tissue culture hood, all manipulations must be carried out in the sterile laminar flow hood. You should remember to turn off the UV light. Ordinarily, it should be on. Turn off the UV light, because if you're working in a laminar flow hood with a UV on, it can also damage your skin and could harm you. Bring materials into the hood, light up the flame of the gas we have talked about, and now you can begin your work. Of course, as I've mentioned earlier, you please flame the caps, the lids of the containers or the tubes, and also the mouth of the tube before you start working. So we have seen how different strategies we can use to sterilize our equipment. In the next module, we'll continue our discussion.